So I welcome all the attendees and the speaker in this science dialogue series. As you all know, this is continuous process and purpose is only to increase knowledge pool of the society, in especially in bioscience system, because this is directly related to my subject. But this is not limited. We are extending in chemistry and other subject also. But chemistry and other subject because that subject is also helping bioscience subject to grow and to understand. So in this series, as you all know, today's speaker is Dr. Jitendra Kumar ji. He's from my native place, Bihar, but he did his MSc from भूल गया.
Jivaji University.
Yes,
Jivaji
Jivaji
University,
University. Yes, sorry.
Gwalior.
Gwalior, and MPhil from Delhi University and PhD from Maharshi Dayanand University. He has also worked in some company as a molecular biology head at Patna. And nowadays he is working since 4 years. He is working as a postdoctoral research fellow at a very prestigious university, Iowa University, USA. That is brief introduction. Common thing is he has also published several research and review paper, presented his research in different conferences. These things are common. As you know, all researchers, they do this. But he is from my place and he is in USA. That is, you know, Gujarati and Punjabis are more in USA. He's from Bihar, so he's also in USA, and since 4 years he's working there. So I welcome Dr. Jitendra ji in our Science Dialogue series, and we are grateful to you that you are ready to deliver lecture. Though this topic is very too specific, but yes, common patterns of all the research are same. How to start, how to hypothesize, material methods. Some of the material methods are common for different research to perform the research. So I request all the participants to interact. You can ask questions during lecture also, so that you will understand. Suppose you are not understanding completely, because I cannot say that new topic you will understand completely. You should understand, but at least you should try to understand, so that that knowledge can be utilized in next lecture, next discourse, next discussion, and next topic, because some of the things are common for all the research. With this brief introduction of Science Dialogue series, you all know, brief introduction of today's speaker. I have said you briefly. You all can interact with him after lecture, so you must attend complete lecture. If and then you can join him on LinkedIn also, Facebook also in future. So now platform is yours, Dr. Jitendra ji. Now you can share your slide.
White wala. Yes, sir. So, good evening to everyone, and thank you, sir, for a nice introduction. So, it's a good opportunity. So, it's a good opportunity for me to share my research experience and my present research work with you guys. So, today's topic is not.
वॉइस कम हो गया थोड़ा सा, जैसे पहले से.
Okay, so 1 minute. It's visible. My
yes, slide is visible. Slide is visible. वॉइस थोड़ा सा बढ़ा दीजिए, बढ़ जाए कहीं से.
Yeah. So voice is okay.
Yes.
Now it's okay.
Yes.
Yeah. So the today's topic is very specific because I am working on this topic. So I don't know how many people are working on post-translational modification. But the main purpose of today's lecture is you guys are new students or some faculty members. They are doing research or they want to do some research in any direction. You can connect your research with the techniques. In USA, what techniques are important and what generally people are using 4, 5, and 6 techniques are very specific. Every lab is using, so that you have some ideas about the USA research, how people are doing research, how you can create your hypothesis, how you can means transfer your ideas into a research project or a grant, and for all this. So I don't want to take more time behind these things. You can ask any question anytime on any slide. Okay, so it's more about the research and experimental part. You can ask in any language, Hindi or English. So it's no any barrier. Don't hesitate with the language. Language is just for a communication. It's not a more means more importance in the science. In USA, people are working without knowing English. So science is important. So I'd like to request to all of you. Okay, you can ask and interact. So my today's topic is p66 mediates SUMO2-induced endothelial dysfunction. So every word here, every word is important. So p66 is a adapter protein, SUMO2 is a small protein, and endothelial dysfunction, it is a endothelial cells. So I will talk about the endothelial cells, how its function is not working because of the adapter protein and because of the small SUMO2 protein. So basically, this endothelial dysfunction causes disease like atherosclerosis, blockage in heart. So I will discuss about that. So here some background related to the post-translational modification. There are different types of post-translational modification because of the polypeptides, because of the chemical groups, because of the other groups. So here my protein is protein of interest is polypeptides protein. So how a small polypeptide proteins targeted some genes like p66SSC is the target protein, and they completely modified its and they changed their function. So first, SUMO2. So it's a eukaryotic proteins and is a so first time it is a single gene reported in yeast. There are four types of SUMOs: SUMO 1, 2, 3, and 4. So based on the sequence homology, SUMO 2 and SUMO 3 are very similar. So most of the time we are considering as a SUMO 2, 3 because only 4, 5 gene sequence difference in these two SUMO 2 and SUMO 3, but the SUMO 1 is 50% sequence difference with the SUMO 2, 3, and SUMO 4 is mostly expressed in different sites like kidney, lymph node, and spleen. So here our goal was SUMO 2, 3 because its similarity is the same and it's means easily overexpressed in the cell lines. Uh, and the newly sumos are generally present in the form of propeptides, and it's converted into the mature when it's the C-terminal glycine motif is removed by the specific sumo isopeptidase like SCNPs. So SCNPs basically cleave the glycine motif at the C-terminal of the sumo, and then the newly sumo and the nascent sumos is converted into the mature protein. So now some basic brief introduction about the p66SSC. So SSCA is a adapter protein and p66 is a member of that protein. So there are three isoforms in SSCA protein, p46, 52, and p66. So based on their structure, 46 and 52 are more similar, only the difference is the cytochrome C. But in the p66, CH2 domain is an additional domain and its length is 583 amino acids. So here our target was p66 because CH2 domain is a keep the difference from the p52 and 46, and it is already reported that CH2 domain lysine 81 is performing the acetylation. It's another type of post-translational modification. So our target was um, p66. And the other reason behind this hypothesis is p66 role is already reported in oxidative stress, and SUMO2 is creating the oxidative stress. So these things was similar. So the hypothesis based on the characteristics of p66 and SUMO2, they both have a role in the oxidative stress. Then is it possible that SUMO2 is creating the oxidative stress via p66SC or not? And it is already reported that p66 have a role in different disease like aging or obesity, atherosclerosis, and diabetes. So yeah, so here I'm sharing some paper from um, these papers helps to generate the idea. This paper is published in PNS by the Kamisi et al. And this paper reported that P66 is a adapter protein and it's a help in the hyperglycemia, induced endothelial dysfunction, and oxidative stress. And here the another paper, it was published in Kim, that reported that SUMO2 regulates vascular endothelial dysfunction. So it was reported that SUMO2 have a role in the vasculature and the vascular biology, and it's reported in the oxidative stress. So our hypothesis was based on that study, and we created a new hypothesis. Okay, is p66 is sumoylated by SUMO2 or not? So here are the some mechanism about the sumylation. So sumylation occurs in the four steps: maturation, activation, conjugation, and ligation. So in the and it's a cyclic process. So once sumo is maturated, nascent sumo is polypeptidally processed by SNPs to remove its C-terminal glycine residue, and it's turned into maturation. Then it's converted into activation with the help of AOS1 UVA2, and forms thioester bond between glycine residue and UVA2. And then it goes to the conjugation, where sumo is then relocated to sumo E2 conjugating enzymes like UBC9. In the left side picture, you can see that free sumo is available, target protein is available, and in the presence of enzyme or ligase, it's converted into sumylated. So free sumo residue is targeted to the lysine of the target protein, and it's sumylated. And again, in the presence of SCNP, the sumo is free because cleaved by the SCNP, and then sumo is available for the next reaction. So ligation occurs, and then again sumylated protein works as a substrate for SCNPs, and it is a reversible and dynamic process which releases the sumo molecule for further reaction. So then, uh, because of the sumylation, what effects occurs on the target protein? So sumylation alter the localization of the modified target protein. Suppose the target protein is present in the cytoplasm, then it will modified by the sumo. So it will translocate it from uh, cytoplasm to mitochondria or in the nucleus, and it will alter the protein reaction. And because of the translocation and alteration of the interaction, it might be change the function of the protein, and it might be change the stability of the protein. So that is the nature of the sumylation and the effect of the sumylation on the target protein. So it also increase the sumylation promotes oxidative stress, and because of the sumylation it impairs endothelial function. So these things are already published in many other research articles, and it causes atherosclerosis. So effect of different sumo isoforms are not always overlapping. So SUMO1 is reported in rescue of cardiac pathology, while increased SUMO2 leads to cardiomyopathy. So these things are already published by the different research groups. So our goal was SUMO2 also affects blood vessels and causes vascular endothelial dysfunction. And with the based on the SUMO2 research data and based on the P66 research data, our hypothesis was whether P66SSC is modified by SUMO2 or not. So I have already mentioned uh, many times this was our hypothesis. So to achieve this hypothesis, we performed some experiments. So I will come on the experiments. So in this picture, I would like to share with you how this hypothesis works and how it will perform and what was the important thing behind this. So here you can see that endothelial cells where P66SSC, so it's based on the published data and the mechanism of these things. So P66 phosphorylated at serine 36, it will isomerized and in the presence of pin1, and then it will translocated into mitochondria, it will produce ROS and it will cause a endothelial dysfunction. So in means cause a disease. So here you can see that the mechanism of the endothelial cells and this based on the hypothesis, and in this picture you can uh, see the outcomes based on our hypothesis. So mechanism is showing SUMO2 P66 stimulated by SUMO2, and we confirm these things to stop the stimulation, and we check after stopping the stimulation it is translocated into mitochondria or not, and what is the effect on the ROS. In the right side picture you can see that. So it will stop the ROS, and by this method we can improve the endothelial functions and we can support the endothelial function after stopping methylation in P66 SSC protein. If it will not stop, then it might be the cause of the endothelial dysfunction and it will cause a disease. So from here, uh, I'm going to share my experiment part. So to confirm the ROS production is happening or not, uh, we perform this experiment in the human cell lines, HUVEC cell lines. It's a human cell line. So in this experiment, we took control and the siRNA P66 SSC. So siRNA P66 SSC basically we decrease the uh, expression of P66 SSC, and here it's a Western blot and the immuno blot. So IB gap DH means it is the overexpression of gap DH. It's a housekeeping genes and it's a control. IBHA we check the uh, HA tag SUMO2, that was our target protein. So we overexpress the SUMO2 in the cell lines. So in the lane, second lane, we are plus and plus. So in the second lane, this is the band. So in the second line, you can see the in the IBHA picture, you can see two bands. So in two SUMO2, we overexpressed in this condition. We overexpressed the SUMO2. So SUMO2 band was confirmed by the antibody HA, and in the fourth lane SUMO2 and the SIP66 SSC. So overexpressed in this lane, overexpressed SUMO2, but P66 effect was gone because we put the SIP66. So we overexpress this, and here this top of the blot IBSSC, in this we can you can see that where we use the SIP66, the effect of the P66 band was almost gone. It's not a 100% knockout. It's a 80%. About siRNA is confirming the it's around 80%, 90% effect was decreased. So here you can see the very light band of P66. So that indicating the effect of P66 is knocked down. And based on this, we perform the ROS production with the help of mitosox fluorescence, and this is the effect. So in the HA SUMO2 is significantly more in control, and when we use the SIP66 in the right side picture, uh, LACG and SUMO2, so no any significant results. That means when This picture is representing when we are knocked down the p66 SSC ROS production was not produced. So based on this, we moved forward and we performed the simulation. So how we will confirm the simulation? So first, from the last experiment, we confirm the ROS production is not occurring when we knock down the P66 effects. Now the question is, P66 is simulated or not? To achieve this objective, we perform the immunoprecipitation assay, and we used SUMO2 and p66 wild type overexpressed in human cell line. So in this in the left side picture, you can see that GAPDH is a housekeeping gene. I have already told. IV flag, this antibody confirmed the overexpression of p66 wild type, because with this gene we add the adapter of flag flag to detect this protein, and IVHA HA adapter is attached with the SUMO2 gene to confirm the SUMO2 protein by using the HA antibody. And in the top two blots, it is a result of IB flag. So in immunoprecipitation with flag, so IP flag means immunoprecipitation with flag. So in this assay, we pull down the flag protein. So what was the flag protein? P66 SSC wild type, because flag was tagged with P66 wild type. So in immunoprecipitation, we pull down the P66 wild type. Pull down means keep separate the p66 wild type protein. Then which protein will express? The HA tag protein only. So after the pull down or removing of the flag tag protein, in the top plot you can see that the only HA tag summulated protein is observed in the lane 3 in the left side picture. So in this condition, p66 wild type and SUMO2, so these multiple bands proteins was showing the simulation of p66 wild type. So with this help of this experiment, we confirm the simulation of p66 wild type. Then it is a true simulation or not. What we do in the right side picture comes in the right side picture. There are you are um, seeing here so many blots. So from the bottom to top I am moving. So the bottom one is a GAPDH uh, housekeeping gene. So it representing equal amount of protein loaded in each lane. Then SENP1, this band after GAPDH, so above blot IB SENP1. This plot showing कि SNP1 is overexpressed in above UBC9, UBC9 is overexpressed, IB flag protein is overexpressed, IVHA HA protein is overexpressed, and then two blots IB flag and HA is the immunoprecipitation flag. So now I'm explaining each conditions. So in the first lane of each pictures, in the first lane I used only P66 wild type. So P66 band only you can see the IB flag means from the bottom to the top fourth blot. So this representing P66 wild type in each condition means in each lane. Then P66 wild type with SUMO2. So second condition in each. So here this band is comes in HA and flag both, because we used the both gene in this condition. Third one with UBC9. So I mentioned you in the previous slides in the background of the story. UBC9 is a conjugating enzymes. So what is the role? It will promote the simulation. And the fourth one I used SENP1. So what is? It's a desumylated enzyme. So it will decrease the simulation. Now I'm coming on the top uh, two blots. So it's a immunoprecipitation blots. So in this again, we pull down the flag tag protein, and we confirm the simulation by using HA tag antibody. So in the lane 2, where only p66 and SUMO2 smylation occurs, the top blot of the right side picture, and the next one, you can see that smylation is increasing with the UVC9, and in the fourth condition, no smylation observed because we use the SENP1. It's a desmylating enzyme. Got it? So these are the overexpression. So with the so this so in the left side picture confirming the smylation of the p66 wild type, and the right side picture is confirming the smylation of the p66 wild type is increasing with the conjugating enzyme, and no any smylation with the desmylating enzymes. Now the question was, okay, is all these things are true? So we confirm these things by using the anacardic acid. So in this picture, we used the anacardic acid. So what is anacardic acid? It's a pharmacological inhibitor of SUMO2. It's an inhibitor of smylation. So here we used in the fourth one condition, we used 2 micromolar anacardic acid, and fifth one 5 micromolar anacardic acid. So, and this is the quantification of this immunoblot. So in the quantification, you can see that, okay, after the immunoprecipitation of lactate protein, the anacardic acid is decreasing the smylation, significantly decreasing the smylation with the 5 micromolar anacardic acid. So the anacardic acid is an inhibitor of SUMO. It's confirming the uh, smylation. So now these things in this we used the different concentration of anacardic acid 0 to 25 and check the effect on the p66. So when we when we increasing the anacardic acid from 0 to 25 with the p66 wild type with the HSOMO2, the p66 SSC p66 amount was increasing with the decreasing the SUMO2. So more p66 in the right one side. You can see that in the picture B, the p66 band is more thicker in comparison with the second lane of this. So it again it was confirmed. Yes, smylation is occurs in the p66 SSC. Now the question was, okay, is it happening in the recombinant protein also? So for this, we confirmed by the mass spectrophotometry, and to confirm this, we run the gel, stain the comassive stain, and confirm the smylation, and then we rerun the SDS page gel and the silver staining, and then we cut the marked band, and we send for the mass spectrophotometry from the silver staining gel. This is the mass spectrophotometer results. So here you can, in the left side, you can see the sequence of the lysine 81, and here in the lysine 81 is marked by the green color. So these things was confirmed by the mass spectrophotometry results. His smylation occurs in p66 at the lysine 81 position. So it was confirmed by the mass. The lysine 81 is responsible for the smylation of the p66. Then the and we confirm the smylation in recombinant protein also in the with the p66 wild type with UVC9. Here we use the smylation kit and confirm the smylation in recombinant protein. Here in the smylation was confirmed in the recombinant protein of p66 wild type and K81 are both. So this is the blue. I have marked with the blue arrow topmost. Some modified p66 SSC in the condition of p66 wild type with UVC9, and no any band in the KT1R with UVC9. Then the question was, its lysine 81 is modified by. So when you uh, mutated the lysine 81 in P66, then it's modified by SUMO2 or not. So in this, for this, we mutated the lysine 81 into arginine. So I have, in the left side picture, you can see the third condition, flag tag P66 SSC K81R. So K81R means lysine 81 is converted into arginine. So in the first condition, we use only SUMO. Second condition, only P66 wild type overexpressed, and third P66 wild type and SUMO2, and fourth one only KT1R, and fifth one KT1R with the SUMO2. So then again, overexpression, check the overexpression results, GAPDH flag, HN flag, and then immunoprecipitation was performed. We pull down the flag tag, tag P66 wild type, and we confirm the SUMylation by using HA antibody. So here you can see that in the condition P66 wild type with SUMO2 is more SUMylated in comparison with the P66 wild type with P66 KT1R with the SUMO2, fourth one. So and the same experiment, so this experiment was performed in HEK293 cells, and the same experiment is repeat with the adenovirus of the SUMO2 P66 wild type and KT1R in performed in the HUVEC cell line. Okay, so in this right side picture, it's a repetition of the left side experiment. So so now the question was, I've already discussed in my previous slide in the mechanism. Okay, after confirmation of SUMylation, the main role, okay, because of the SUMylation, serine 30 phosphorylation is occurs on the P66 or not, means because of the SUMylation, any effects on the phosphorylation of the P66 or not, because it is already reported and published that phosphorylation of serine 36 in P66, it helps in the translocation of P66 into the mitochondria. So here we performed this experiment and we confirmed that in this figure A is a immunoblot of P66 SSC serine 36 phosphorylation. So P66 SSC S36 means we use the specific antibody to detect the serine 36 phosphorylation. So you can see that the serine 36 6 phosphorylation is increasing in the P66 wild type SUMO2 in comparison with the only P66 wild type, and it's a significantly increased. So that this result indicates, so because of the SUMylation of P66 SSC, serine 36 phosphorylation is increasing. And again, this experiment is confirming in the with the um, in the in the HUBEC cell lines. So we generally replicated the experiment in HEK293 and HUBEC both. So in the both cell lines, we observed the same results, and here also the serine 36 was increasing with the SUMylation of P66 wild type. So this is the immunoblot results. In the right side, quantification of the band of the uh, uh, immunoblot. So for the quantification, I used the graph prism software. So after the confirmation of the phosphorylation, the question is, hello, hello.
हाँ सुन रहे हैं
Yeah. So if you have anyone have any doubt, then please let me know and ask about the experiment and the conditions and anything. और कहीं पे अगर नहीं समझ में आ रहा है तो आप पूछ सकते हो कि मुझे आप लोग दिख नहीं रहे हो, तो मैं अपने
हाँ
फ्लो में
लेकिन
कोशिश
हम
कर
आप
रहा हूँ
दिख रहे
धीरे
हैं
धीरे.
सबको
अच्छा,
आप
ओके.
सबको
तो
दिख रहे हैं
हाँ, तो फ्लो, माय फ्लो इज ओके, नो
यस
टू
यस
फास्ट, नो
यस
टू
यस
स्लो.
कुछ पार्टिसिपेंट्स जो है ना वो निकल गए, उनको स्लो लग रहा है, बट क्या है
ओके,
सबके
ओके.
लिए तो है नहीं। प्रणव सर भी आ गए हैं।
हाँ, राइट. ओके सर, गुड मॉर्निंग, गुड इवनिंग.
Good evening. Good evening.
Yeah, yeah. So after, after the confirmation of the phosphorylation of serine 36, the main goal was to check the serine phosphorylation is occurs, then P66 is translocated into mitochondria or not. So to achieve this objective, we fractionated the cells and we checked the P66 level in the cytoplasm and in the mitochondria and the nucleus. So here this blot is confirming the translocation of the uh, P66. So yeah, so here P66 wild type, and here again we confirm the phosphorylation of serine 36 with KT1R. So here you can see that the serine 36 level was significantly decreased in the with the KT1R, and this is the quantification of this blot. So it's a control gap DH, overexpression of HA gene, overexpression of flag, and then the it's a confirming the phosphoserine 36. So yeah, so next level was to confirm the level of P66 in the mitochondrial fractionation. So this blot showing these things, and that was normalized with the COX-4 antibody. COX-4 antibody is indicating the mitochondrial uh, fraction. So here we use the different MOI of SUMO2. So when we increasing the MOI of the SUMO2 virus, adenovirus of SUMO2, it was HA tagged. The P66 level was increasing in the mitochondrial fraction. So the lower in the figure A, lower membrane was IB gap DH. It's a housekeeping gene. I've already mentioned it's a IB flag, and then IBHA, the overexpression of SUMO2. So we increasing the MOI SUMO2, the SUMO2 overexpression was increasing, and these two top band was indicating in the mitochondrial fractionation, and it was normalized by COX-4. So the means flag is increasing in the blot means P66 was increasing, and it's a significant. So right side in the figure B, it's a quantification of the P66. So the flag tag was increasing with the increasing of the MOI of the SUMO2 virus. So it indicates the translocation of P66 into the mitochondria, and it was changing with the amount of the SUMO2 virus. Then we checked with the P66KT1R translocation change with the P66KT1R. So when we used the mutated P66 wild type in comparison with the wild type P66 wild type with the SUMO2, the translocation was decreasing. So because what is the hypothesis? So P66 was mutated at lysine 81. So the SUMO2 target lysine is not present in the P66, so not more sumoylated, so not more translocated in the mitochondria. So in the condition P66 wild type in the figure C, P66 wild type is SUMO2. The band was thicker in the flag in the mitochondrial condition, and with the KT1R it was very less, very means faint band. So when we quantify these things, so quantification results relatively abundance of P66 was showing in the mitochondria. You can see the quantification of figure D. So that indicates yes, after the mutation of lysine 81 in P66, sumoylated is compromised, and because of that the less amount of P66 is translocated, so we can control the translocation of P66. Then what will happen after the controlling of translocation of P66? It will not available uh, in the mitochondria, then it will not produce the ROS. In the first figure I show you the ROS production was because of the P66. So we are control the P66 translocation into the mitochondria from the cytoplasm to mitochondria. So it is controlling the ROS production. So why it is important? Because ROS will produce more, then it will affect the endothelial function. It will convert the endothelial, it will cause the endothelial dysfunction, and because of that, we can move cause for the atherosclerosis disease. So we can control these things. So because of that, its significance is important. So then the question was, so this experiment was in vitro experiment, so now we confirm these things in the in vivo. So for this we used in the mice, so we use the different types of mice and dissect the aorta of the mice, open heart open heart cavity of the mice, and then we isolate the aorta. So this is the whole aorta in the left side, heart to the bottom means till the around the lower abdomen part. We isolated the entire aorta, make a small ring, and mount in this viromyography chamber, and we perform the vascular reactivity assay. So this experiment is important to confirm the relaxation and contraction in the aortic ring. So, and we confirm the mice. So it's wild type mice, homo mice, and het mice. We confirmed by the genotyping. So these are the sequencing, and this plot figure B confirming the wild type, homo, and het mice. So we use the wild type and homo mice. So and these are the immunoblot. So that ring was the protein was isolated from that viromyography used ring, and we confirmed the HA and the GAPDH in that ring, because we use the SUMO virus to infect that ring. So here are the result of the viromyography. So with this experiment, we confirm the p66 somylation impairs the endothelial dependent vasorelaxation. So we checked the endothelial dependent and endothelium independent experiment in the wild type p66 mice and KT1R mice. So I'm explaining this experiment. So in the wild type mice, in the both wild type and KT1R mice, we isolated the Arctic ring, and we infected with the HSUMO2. So the purpose was, okay, is there P66 was present in the endothelials of the mice aorta. SUMO2 was there, and produce the sumylation with the p66 wild type in the mice. So here in the figure A, we can see that the vasorelaxation was significantly delayed in the wild type mice, and when we mutated, we used in the knockdown of the p66k81r mice aortic ring, the no any vascular delayed relaxation in this mice in the endothelium dependent, and no any difference in the wild type and KT1R mice in the endothelium independent independent. So that results significantly and strongly present endothelium delayed relaxation is because of the present in the significantly increasing in the wild type mice in comparison with the wild type mice. So again, it's proof these things. Okay, if we mutated the lysine 81, then it will not present, not available for the SUMO2, and when the SUMO2 was stopped, it will improve the endothelial functions. So this experiment is performed in the mice. So both way, in vitro and in vivo, we confirmed these things. So SUMO2 is occurs via P66, and it is responsible for the endothelial dysfunction. So here again, we checked these things in the prevent the it's hyperlipidemia induced endothelial dysfunction and oxidative stress. So in figure A, we use the OLDL, and we confirm the immunoglot SUMO2 and 3. So with the OLDL, SUMO2 overexpression was increased. It was showing that, okay, the oxidized LDL is responsible and increasing the SUMO2-3 level. And these again, we performed the viromyography experiment with the LDLR hyphidite mice and LDLR. So LDLR knockout mice is a uh, animal model for atherosclerosis disease. So when you are giving the high diet of LDLR mice, the atherosclerosis disease will cause in the LDLR knockout mice. So we use the high diet mice. And in figure C, you can see that it's significantly delayed relaxation in the LDLR high-fi diet mice in comparison with the blue, so red line was indicating the LDLR high-fi diet mice, and the blue line is representing the LDLR mouse when bred with the p66 K81R knock-in mice. So these mice was means the mutated of the lysine 81, and it was uh, uh, crossbreed with the uh, atherosclerosis uh, mice. The animal model of the atherosclerosis mice means LDLR. Then the delayed relaxation was decreasing, and it's equivalent to the wild type means the control mice. So this results indicating that, okay, with the knockdown of p66 or the mutation of the p66, it will improve the atherosclerosis disease. So based on these risks, and this is the quantification, it was means significantly improving, and these things was not happened in the and the figure B is confirming the serum cholesterol level. So in the comparison with the wild type and LDLR high-fi diet and LDLR p66K1R mice, it was significantly serum cholesterol level was significantly changing with the LDLR knockout mice. So the based on these data, again it was proven that after the mutation of p66, it will improve the endothelial function. So the overall conclusion of my study. So this figure is presenting the conclusion of my study. So when we stop the stimulation of the p66 SSC, it will not move into the mitochondria. It will not meet with the cytochrome C, and it will so ROS will not produce, and it will help in the uh, improvement of the endothelial function. It will stop the endothelial dysfunction, and because of that we can control the atherosclerosis disease. So the significance of this study was offers valuable insights into the role of post-translational modifiers, especially SUMO at lysine 81, and its impact on endothelial function through reactive oxygen species. So these are the acknowledgements. So my Kumar Lab member of the Kumar Lab, and my mentor Santosh Kumar. I would like to thank for the mass spectrometry, Dr. Vikas and NIH for the fund, and the viral core department, the University of IBA, and the genome editing core facility, University of IBA. And thank you all of you for your
थैंक यू बहुत अच्छा से
patience.
बहुत स्मूथली आपने एकदम
And
बताया जो